everyone, my name is Tana and I will be presenting the introduction for our topic, comparison of extraction techniques for phenolic compounds in Camellia sinensis. Phenolic compounds are among the major bioactive compounds in Camellia sinensis, contributing to its strong antioxidant, anti-inflammatory, antimicrobial and overall healthy promoting properties. Because these compounds are sensitive to heat, light and oxidation, the efficiency of their extraction can vary greatly depending on the technique used. Conventional methods such as infusion, maceration and solvent extraction remain widely practiced due to their simplicity. However, they typically require long extraction time, high solvent volumes and will lead to terminal depression or reduced recovery of decayed phenolics. In contrast, modern extraction technologies such as microwave-assisted extractives (MEE) and ultrasound-assisted extractive (UAE) are much a greener and more efficient alternative. This method ensures mass transfer, shorten extraction during and after, yield higher phenolic concentration while using less solvent. Despite growing interest, past studies tend to investigate only individual methods or compare only conventional or only modern techniques. There is limited research offering a direct systematic comparison between conventional and modern extraction approaches specifically for Camellia sinensis. Since phenolic compounds differ greatly in structure, stability, and biological activity, optimizing extraction condition is crucial to minimize both yield and functional quality. Understanding how each extraction and both influences phenolic recovery is therefore essential for providing high-quality tree extracts for application in foods, beverage, and natural formulations.
My name is NURULAIN BINTI JOHARI and my metric number is J23A0740. Our research is titled Comparison of Extraction Technique for Phenolic Compound in Camellia sinensis. This section identifies the key research problems that make this comparative study critically necessary. A literature analysis reveals one fundamental problem: the methodologies of existing studies are highly inconsistent. This problem leads to two specific problems. First, there is no direct comparison between conventional and modern techniques, specifically for tea. Second, existing modern studies are very limited. They only compare MAE versus UAE among themselves or focus on other Camellia species. The direct consequence of this problem is no systematic evaluation to determine which technique is most efficient for Camellia sinensis. Most critically, our current extraction study ignore the biochemically perspective. That is, performance is not linked to the actual molecular forms and bioactivity, even though that is the ultimate goal. Therefore, the field suffers from inconsistent methods, incomplete comparisons, and a neglect of biochemistry. This is the exact problem our study will fill. Thank you.
My name is SADONA and my metric number is J23A0472, and I'm taking the objective part. So our first objective is to determine the phenolic yields obtained from different extraction techniques. This means that we want to measure how much phenolic compounds each method can release from the tea leaves. Different extraction technique method, which is like infusions, macerations, micro-assisted extractions, and ultrasound-assisted extractions, can produce different amounts because they break the plant structure differently, and this object focuses on quantify the phenolic concentrations so we can see which method is naturally more productive. And our second objective is to evaluate the extraction's efficiency, selectivity, and practicality of each method. Here, the efficiency means how fast and how efficiently the method extracts phenolic compounds, and also the selectivity refers to whether the techniques can target phenolic compounds without extracting too many unwanted components. And practicality include factors like time, solvent usage, and simplicity. This objective actually helps us understand the overall performance of each extraction technique beyond just the yields. And our last objective, which is the third, is to compare the phenolic profile produced by each extraction method. Phenolic compounds are not all the same. They are very in structure and bioactivity. So even if two methods produce similar yields, the type of phenolic extracted may be different. By comparing these profiles, we aim to identify which techniques give the most complete or most desirable phenolic compound and phenolic compositions, so that is more standardized extraction methods can be recommended for tea phenolics. Especially we know that there are some maybe hard sensitive phenolic compounds, so that's why we do know which type extraction method can actually help us to get the one compounds that we actually needed. And that's all from me.
This hypothesis suggests that modern extraction techniques such as microwave assisted extraction (MAE), ultrasound assisted extraction (UAE), or supercritical fluid extraction (SFE). This extraction will outperform conventional methods like maceration or oxalate extraction when extracting phenolic compound from Camellia sinensis. Modern techniques use advanced mechanisms such as rapid heating, cell wall disruption, and improved solvent penetration, which are expected to release more phenolic from plant tissue. Because of this, they should produce higher phenolic yield, meaning more phenolic compounds are extracted per unit of plant material. They also offer better extraction efficiency, often requiring less time, lower solvent volumes, and providing deeper penetration into plant cells. Finally, this method may generate extract with more diverse or intense phenolic profiles, as they can liberate compounds that conventional method might miss or degrade. Overall, the hypothesis predicts that modern method will deliver superior quantity, quality, and efficiency in phenolic extraction.
Assalamualaikum and hi, my name is DAMIRAH, and today I will present the detailed methodology for our study. First, I will explain sample preparation. To eliminate variability, we source leaf from a single batch, ensuring consistent age and foam condition. The leaf are first cleaned and then freeze-dried, or a process called lyophilization. This process gently removes moisture under a vacuum, preserving thermosensitive compound that could be damaged by conventional drying. The lyophilized leaf are then ground in a laboratory mill to achieve a uniform powder, creating a consistent starting material for all extraction. Next, I will explain extraction methods. We compare two conventional and two modern techniques, all designed to extract phenolics into a solvent. For the conventional techniques, we have infusion and maceration. For infusion, the powder is soaked in boiling distilled water in a temperature-controlled water bath for 10 to 30 minutes, simulating traditional tea preparation. The extract is then immediately cooled, filtered, and dried. For the maceration, the powder is soaked in 70% ethanol at room temperature for 24 hours with occasional agitation. This is the standard prolonged solvent extraction. The extract is then filtered and dried. For the modern techniques, we have microwave assisted extraction or MAE. The powder is mixed with 70% ethanol in sealed Teflon vessel. The microwave energy rapidly heats the solvent to between 50 and 150 Celsius, creating intense pressure for 10 to 15 minutes. This disrupts the blend cells efficiently. For the ultrasound assisted extraction or UAE, the powder in the 70% ethanol is placed in an ultrasonic bed at 40 kHz. The gravitation bubbles formed by sound wave physically rupture the cell walls. The temperature is carefully maintained between 40 and 60 Celsius for 30 to 45 minutes to prevent thermal degradation. All the resulting extract from these two methods are cooled, filtered, and dried to a constant way for analysis. So, I will explain the analysis. We evaluate the extract from the three critical angles. First, yield and total phenolic content or TPC. We calculate the dry extract yield. Then, using the following calculator method, we quantify the total concentration of phenolic compounds expressed as milligram of gallic acid equivalent per gram. Second, phenolic profile. We use high-performance liquid chromatography or HPLC to identify and quantify specific amount like catechins, gallic acid, and flavonoids. This reveals not just how much but what is extracted by each method. Third, process efficiency. We record the extraction time, temperature, and solvent volume used for each technique. This data allow us to evaluate the energy efficiency and grain credentials of each method for potential industrial application. And lastly, statistical validation. To confirm our findings are scientifically sound and not due to chance, we use one-way ANOVA to determine if the differences in the mean TPC and the phenolic yields among the four techniques groups are statistically significant. If ANOVA shows significance, we then perform the Tukes-Horner's significant differences was Holt test. This test pinpoint exactly which specific pairs of the techniques. For example, maceration versus microwave are significantly different from one another. So with this methodology allows us to systematically determine not only which techniques recovers the highest phenolic yields, but also how the phenolic profile differs and which method offers the best balance of efficiency, versatility, and compound preservation. That's all from me. Thank you.